welcome back to Nigham's BioNinja. In this video, we are going to cover SLO based MCQs and scenario based short questions with answers from first year biology, National Book Foundation, chapter 1, cell structure and function, 1.1 techniques used in cell biology, and this is the third part of this video series. These are the techniques which are covered in the textbook content, and out of these techniques we will be covering electrophoresis and spectrophotometry related MCQs and scenario based short questions in this video. We have a total of 20 MCQs and 5 scenario based short questions in this video, so without further delay let's get started with the MCQs. Number 1. Which of the following types of spectrophotometry is used to determine the concentration of a protein sample? A. Visible spectrophotometry. B. UV visible spectrophotometry. C. Infrared spectrophotometry. D. X-ray crystallography. The correct answer is B. UV visible spectrophotometry. Number 2. Which of the following ratios is used to determine the purity of a DNA sample using spectrophotometry? A. Absorbance at 260 nanometer divided by absorbance at 280 nanometer. B. Absorbance at 280 nanometer divided by absorbance at 260 nanometer. C. Absorbance at 220 nanometer divided by absorbance at 280 nanometer. D. Absorbance at 280 nanometer divided by absorbance at 220 nanometers. So the correct answer is option A. Absorbance at 260 nanometer divided by absorbance at 280 nanometers. Number 3. Which of the following types of spectrophotometry is used to measure the growth rate of bacterial cultures? A. Visible spectrophotometry. B. UV visible spectrophotometry. C. Turbidometry or OD measurement. D. Fluorescence spectrophotometry. The correct answer is C. Turbidometry or OD measurement. Number 4. Which of the following types of spectrophotometry is used to determine the concentration of a solution of a colored compound? A. Visible spectrophotometry. B. UV visible spectrophotometry. C. Infrared spectrophotometry. D. Fluorescence spectrophotometry. The correct answer is A. Visible spectrophotometry. Number 5. Which of the following types of spectrophotometry can be used to measure the activity of an enzyme? A. Visible spectrophotometry. B. UV visible spectrophotometry. C. Infrared spectrophotometry. D. Both A and B. The correct answer is D. Both A and B. Number 6. A researcher wants to determine the concentration of a protein sample using spectrophotometry. Which of the following wavelengths should the researcher use? A. 280 nanometer. B. 220 nanometers. C. 540 nanometer. And D. 620 nanometers. So the correct answer is A. 280 nanometer. Number 7. A student is conducting an experiment to determine the concentration of a DNA sample using spectrophotometry. After measuring the absorbance at 260 nanometer and 280 nanometer, the student calculates a ratio of 1.8. What does this ratio indicate? A. The DNA sample is pure. B. The DNA sample is contaminated with protein. C. The DNA sample is contaminated with RNA. D. The DNA sample is contaminated with salt. So the correct answer is A. The DNA sample is pure. Number 8. A researcher wants to determine the concentration of a solution of colored compound using spectrophotometry. Which of the following wavelengths should the researcher use? A. 260 nanometer. B. 280 nanometer. C. 540 nanometer. D. 620 nanometers. The correct answer is C. 540 nanometers. Number 9. When measuring absorbance using spectrophotometry, which of the following is correct equation? A. Absorbance is equal to sample intensity divided by reference intensity. B. Absorbance is equal to reference intensity divided by sample intensity. C. Absorbance is equal to sample intensity minus reference intensity. And D. Absorbance is equal to reference intensity minus sample intensity. The correct answer is A. Absorbance is equal to sample intensity divided by reference intensity. Number 10. Which of the following types of molecules is most commonly measured using spectrophotometry? A. Lipids. B. Proteins. C. Nucleic acids. And D. Carbohydrates. The correct answer is C. Nucleic acids. Number 11. Which of the following is the correct order of steps in gel electrophoresis? A. Load sample. Apply electric field. stain gel. B. Apply electric field, then load sample and then stain gel. C. Load sample, then stain gel and then apply electric field. Or D. Stain gel, then apply electric field and then load sample. So the correct answer is C. Load sample, then stain gel and then apply electric field. Number 12. Which of the following types of molecules can be separated using electrophoresis? A. Proteins, B. DNA, C. RNA and D. All of the above. The correct answer is D. All of the above. Number 13. Which of the following is correct definition of electrophoresis? A. A technique used to measure the concentration of a sample using light absorption. B. A technique used to separate molecules based on charge and size using an electric field. C. A technique used to amplify a specific DNA sequence using polymerase chain reaction. Or D. A technique used to visualize the location of a specific protein in a cell using antibodies. So the correct answer is B. A technique which is used to separate molecules based on their charge and size using an electric field. Number 14. A scientist wants to determine the concentration of a particular compound in a water sample. Which of the following techniques would be most suitable for this purpose? A. Microscopy, B. Chromatography, C. Spectrophotometry, D. Tissue culture. The correct answer is C. Spectrophotometry. Number 15. Which of the following is the correct definition of SDS page? A. A type of electrophoresis used to separate nucleic acids based on the size. B. A type of electrophoresis used to separate proteins based on charge. C. A type of electrophoresis used to amplify a specific DNA sequence. Or D. A type of electrophoresis used to visualize the location of a specific protein in a cell using antibodies. The correct answer is B. A type of electrophoresis which is used to separate proteins based on charge. Number 16. A student is conducting an experiment to determine the concentration of a red food dye in different beverages. Which of the following steps would be appropriate in the spectrophotometric analysis? A. Prepare a microscope slide with the food dye samples. B. Use chromatography to separate the components of the food dye. C. Measure the absorbance of the food dye samples at a specific wavelength. Or D. Grow the food dye samples in a tissue culture. The correct answer is C. Measure the absorbance of the food dye samples at the specific wavelength. Number 17. A scientist wants to analyze DNA fragments obtained from a crime scene to determine the suspect. Which of the following techniques would be most appropriate for this purpose? A. Spectrophotometry, B. Microscopy, C. Chromatography and D. Electrophoresis. So the correct answer is D. Electrophoresis. Number 18. A biology student is conducting an experiment to determine the genetic variation among individuals of a particular species. Which of the following electrophoresis techniques would be most suitable for this purpose? A. Gel electrophoresis of DNA fragments. B. SDS page for protein analysis. C. Capillary electrophoresis for nucleotide sequencing. And D. None of the above. So the correct answer is A. Gel electrophoresis of DNA fragments. Number 19. A pharmaceutical researcher is developing a new drug and wants to assess the purity of the synthesized compound. Which electrophoresis technique would be most suitable for this purpose? A. Gel electrophoresis of DNA fragments. B. SDS page for protein analysis. C. Capillary electrophoresis. D. None of the above. The correct answer is none of the above. Number 20. A microbiology student is studying the bacterial protein expression. Which of the following electrophoresis techniques would be most suitable for separating bacterial proteins? A. Agarose gel electrophoresis. B. SDS page for protein analysis. C. Two-dimensional electrophoresis. And D. Capillary electrophoresis. The correct answer is B. SDS page for protein analysis. That is also far from the MCQs covered from these topics. I hope that you have learned a lot. Now we will move to SLO based or scenario based short questions from these topics. Number 1. Nigham wants to analyze the DNA of a suspect in a criminal case. What type of electrophoresis technique can he use? The answer to this will be Nigham can use gel electrophoresis to analyze the DNA of the suspect in a criminal case. In gel electrophoresis, DNA fragments are separated based on their size and charge by applying an electric field to a gel matrix. Number 2. Aisha wants to analyze the protein content of a food sample. What type of electrophoresis technique can he use? The answer to this will be Aisha can use protein gel electrophoresis to analyze the protein content of a food sample. In protein gel electrophoresis, proteins are separated based on their size and charge by applying an electric field to a gel matrix. Number 3. Zainab wants to analyze RNA transcripts of a gene in different tissues. What type of gel electrophoresis technique can she use? The answer to this will be Aisha can use northern blotting electrophoresis to analyze RNA transcripts of a gene in different tissues. In northern blotting, RNA molecules are separated based on their size by applying an electric field to a gel matrix, then transferred to a membrane and probed with a labeled RNA probe to detect the target RNA. Number 4. Aisha wants to determine the concentration of a protein sample. What type of spectrophotometry can she use? The answer to this will be Aisha can use UV visible spectrophotometry to determine the concentration of a protein sample. In UV visible spectrophotometry, light of a specific wavelength is passed through a sample and the amount of light absorbed is measured. The amount of absorption is proportional to the concentration of the sample. Number 5. Bilal wants to determine the purity of a DNA sample. What type of spectrophotometry can he use? The answer to this will be Bilal can use UV visible spectrophotometry to determine the purity of a DNA sample. In UV visible spectrophotometry, light of a specific wavelength is passed through a sample and the amount of light absorbed is measured. The purity of DNA can be determined by the ratio of absorbance at 260 nanometer and 280 nanometer. Since 260 nanometer gives the absorbance of DNA and 280 nanometer gives the amount or absorbance of protein. So this defines the purity of a DNA sample. That's all for today from these two contents of techniques in cell biology. I hope that you have learned enough. Please don't forget to subscribe to my channel. Until next time, this is Nigham's BioNinja signing off.